At the end of this course, we expect that you should be able to classify carbohydrates, classify nutrients into carbohydrates, lipids, proteins, and classify them as macronutrients. We expect also that you have a little bit of knowledge about the chemical structure and properties of macronutrients, that you'll also be able to identify and compare local and continental food sources of carbohydrates, lipids, and proteins. Also, we expect that you're able to identify carbohydrates and lipids as energy sources, and highlight the body requirements of carbohydrates, lipids, and proteins. Okay, so we move on into carbohydrates, which is the first module. And going into that, you should be able to see, to classify carbohydrates based on their structure. So whether they are monosaccharides, disaccharides, or polysaccharides. You should be able to classify carbohydrates also based on their reducing ability. So whether they are reducing sugars or complex sugars. You should be able to also identify common food sources of carbohydrates. So think about it, the food sources around you, the foods you normally consume. Yeah, you should be able to do that. And of course, for your age group, at least you should be able to know the bodily requirements of carbohydrates that is required for making healthy food, feeding or eating choices, and for maintaining normal body weight depending on your physical activity level. Okay, so carbohydrate proteins and lipids are referred to as macronutrients, and we call them macronutrients specifically because they are required in large quantities by the body. And this is opposed to vitamins and minerals, which are called micronutrients, specifically because they are required in small quantities by the body. So what are the functions of carbohydrates? Well, let's begin with energy, which is the very obvious reason. So carbohydrates are responsible for energy production. So we know that in the body, energy is stored as ATP, and carbohydrates basically in our foods in the form of glucose is responsible for the ATP production. So we understand the details. For those of us that have done biochemistry, we understand how energy is generated from glucose. We don't have to go into all the complexities of it, but it's just good to understand that glucose, an example of carbohydrate, is responsible as the primary energy source. Secondly, carbohydrates are important storage for energy as well. So think about the animal cells where carbohydrates or where energy is stored as glycogen. So carbohydrates provide structural functions. So for example, if you think about your chitin, in exoskeletons, for example, chitin is a very good example of structures and the structural function of carbohydrates. But if you also think about cell membranes, carbohydrates play an important role for signaling. So how cells communicate with each other. So imagine you have different cells, so on the cell membrane, cells usually do communicate, and you find certain structures, on cell membranes. So for cell 1, for example, cell 2, for example here, these structures on cell membranes, play a critical role that we call signaling, and those structures are largely carbohydrates. Carbohydrates also play a very important role in fibers. And what is the role of fibers? Fibers play an important role in digestion, in aiding digestion. They facilitate the proper digestion of foods. Oftentimes, for example, you hear that doctors recommend, okay, take a lot of foods in fiber so that you can prevent indigestion. Fibers are gotten from some fruits, for example, like that part of oranges that you like to eat, the inner part, for example. Vegetables are also very good sources of fibers, but they aid in bowel movement, and in that process, make digestion easier. But those components in fiber are carbohydrates. And next, if you think about your nucleotides, so nucleotides are very, very important sources or examples of carbohydrates. So think about a nucleotide. So basically a ribose sugar. And if you think about your DNA, you know that your DNA is made up of a ribose sugar, a base, and a phosphate group. So yeah, that ribose is obviously carbohydrate. And that component you find in your cells, in the chromosomes and all that, they are important sources of carbohydrates. So this ribose sugar forms the backbone of the DNA. Carbohydrates also play an important role in detoxification in the liver. Okay, so these are some of the important functions of carbohydrates. Next, we move to the classification of carbohydrates. So now, it's important to see carbohydrates as simple and complex. An example of simple carbohydrates are monosaccharides and disaccharides. And if you look at monosaccharides, examples are glucose, fructose, galactose. So we know glucose, we know fructose, we know galactose. I want you to start thinking about sources of glucose and fructose. I'm not going to tell you that for now, but for galactose, well, milk is an example of a source where you can find galactose. But these three, the glucose, fructose, and galactose are monosaccharides, whereas for disaccharides you have maltose, lactose, and sucrose. As we proceed, we'll see exactly how they are disaccharides and monosaccharides. For polysaccharides, we have a very good example as starch, fiber, and glycogen. We already talked about glycogen, where you find them as storage, energy storage in animal cells. We also talked about fibers and starches. We'll go into details about them later on. So, an important thing to note is that, well, sugars, monosaccharides in this case can be classified as exose sugar. So exose meaning that it has 6 carbon atoms, but also you can classify sugars, monosaccharide sugars, as pentoses, like the ribose that we considered earlier on. Remember, the backbone of DNA. And the important dietary monosaccharides are the ones we considered earlier on: glucose, fructose, galactose. Okay, so talking about carbohydrates and their number of carbon, or the carbon chain, we can also have carbohydrates, monosaccharides, classified as aldoses or ketoses. So aldoses are those monosaccharides or those sugars that have an aldehyde group. So take, for example, you remember the aldehyde group, whereas ketoses are those monosaccharide or sugars that have the ketone group. So you have aldehyde group as aldoses and ketone as ketoses. But we also have some other classifications. So for example, the triose sugars are sugars that are 3 carbon, tetrose sugars 4 carbon, pentose 5 carbon, hexose 6 carbon sugars. Okay, so it's quite important to understand these structures. So the reason why we're going into details about these structures is, well, it helps our brain to basically understand these concepts. So when I'm talking about hexoses, I'm talking about bonds, for example, we'll come to that. The names of the sugars, the names of the bonds. Yes, it's something that you don't need to cram them. And understanding what is going on at a structural level can be very, very helpful. So the first concept that we're going to learn with regards to the structures or the chemistry of monosaccharides is cyclization. So what exactly happens in cyclization? In cyclization, the open chain form of glucose, for example, a monosaccharide, is converted to the cyclic form of the monosaccharide. And how does this happen? It happens basically when the aldehyde group on carbon 1 interacts with the alcohol group on carbon 5. Okay, so you have a kind of like electron donation, nucleophilic reaction going on there. But basically, it's that interaction that leads to the conversion of the acyclic form to the cyclic form, and it's called cyclization. Okay, so in this example, we can see glucose, let's call it 1 here, and we can see galactose as 2 examples. Now, you have different chemical configuration or structural formula for glucose. Okay, sometimes, for example, from what you can see in 1, yeah, the carbon atoms are not shown, but here the carbon atoms show. So when you don't see the carbon atoms, well, it doesn't mean they're not there. So they're just different forms in which you can write glucose and galactose. But the important thing here that I want to draw your attention to is let's label the carbon. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So if you take a careful look at carbon 4 here, so if you look at carbon 4 here, you see that the hydrogen of carbon 4 compared to galactose has been rotated. So it's like a flip around. And this is an important concept that we need to take note of. And it's a concept that we call epimerism. So epimerism, it's basically when you look at glucose and galactose and you look at the carbon 4 of glucose, you can see that carbon 4 here has the H on the left side and carbon 4 here for galactose as the H on the right side. So we call this concept epimerism, and we'll see it as we proceed. Another thing to look at with regards to the chemical classification of glucose, galactose, and fructose is that glucose and galactose are basically aldehydes. So you can see the C double bond OH group. But for fructose, fructose is a ketone. So if you look at it, you see that this is a carbon, so it's a C double bond O group. So it's good to take note of that. And one thing here is that in this configuration, this is the acyclic configuration, and this is the cyclic configuration for glucose, galactose, and fructose. Okay, now we look at the concept of anomerism. Now, anomerism is an important concept to discuss, and due to cyclization, an additional asymmetric center is created at carbon 1, and this leads to the formation of two isomers. So, namely alpha D glucopyranose, which we popularly call alpha D glucose, and beta D glucopyranose, which we popularly call beta D glucose. Okay, so basically what I want to do here is to explain how anomerism leads to the formation of alpha D glucose and beta D glucose beginning with D glucose. Okay, so now what happens is that as a result of cyclization, which I explained earlier on, between carbon 1 and of course carbon 5, you have a situation in which the H and the OH group on carbon 1, which we normally call anomeric carbon, flips. So basically what is happening is that there is a kind of flip going on here. So in the alpha D glucose, the H is up and the OH is down. But as a result of anomerism, you have more like a flip on carbon 1. Remember, I called it the anomeric carbon. So what happens as a result is that the OH, flips up and the H goes down. But now I'm not going to go into the complicated chemistry of it, but the important thing that you need to know is that this phenomenon results in the formation of alpha D glucose and beta D glucose. Okay, we will see how this plays out. For example, when you begin to see different molecules of glucose bonding together, but we will look at that subsequently. Okay, earlier on we talked about the concept of epimerism. So epimerism occur basically if you're comparing glucose and galactose. Epimerism occur when you have a flip between the hydrogen and the hydroxyl group of carbon 4. So if you look at glucose on carbon 4, the hydrogen group is on the left, the hydroxyl group is on the right, and in galactose on carbon 4, there is a flip. So basically that is the only thing that differentiates glucose from galactose. And as a result of that, we say that glucose or galactose is a C4 epimer of glucose. Okay, but basically what happens resulting in that phenomenon is called epimerism. Okay, having explained that concept, we look into how chemical bonds are formed in disaccharides. Don't forget that earlier on we said that disaccharides, a good example of disaccharides is maltose. And what is happening in maltose? One sugar molecule or one glucose molecule combines with another glucose molecule, and for that combination to occur, there is a bond basically called the glycosidic bond, and it is that bond that holds the disaccharides together, and we call that bond alpha 1,4 glycosidic bond. Okay, this is the importance of understanding the structure. It kind of like helps you mentally to picture exactly what is happening. So if you look at the case of maltose, if you're going to look at the carbon atom, you have carbon 1 here, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So in this order one, you have your carbon 1, 2, 3, 4, 5, 6. So the bonding specifically occur between carbon 1 of one glucose molecule and carbon 4 of the other glucose molecule. Hence, we call that bond 1,4 glycosidic bond. Pretty straightforward. Now, when you have different combinations of monosaccharide molecules, like glucose combining together, yes, if you have about up to 9 monosaccharide molecules, you can still call that compound an oligosaccharide. But generally, when there are more than 12 and all that, it becomes a polysaccharide. So let's consider the polysaccharide starch and the polysaccharide glycogen. So what happens in the case of starch and in the case of glycogen? In starch, you notice essentially that the bonds between the individual monosaccharide molecules, so let's see, for example, this is glucose 1, glucose 2, glucose 3, glucose 4. So you notice that it's 1,4 glycosidic bond, 1,4 glycosidic bond, 1,4 glycosidic bonds. And it's a very, very important structural detail that you notice in starch. It's a completely straight polysaccharide. But if you move on to glycogen, you see that you have 1,4 glycosidic bonds, but in addition, you have 1,6 glycosidic bonds. And as a result of the 1,6 glycosidic bonds, you notice that the structure of glycogen, for example, the storage of carbohydrates in animals is a little bit different. So it branches. So not only do you have this straight chain structure, but you also have a branched chain structure. And that is unique to glycogen compared to starch. But what makes that really possible is as a result of the alpha 1,6 glycosidic bonds that you have in glycogen. Okay, so well, in this table you have the major dietary carbohydrates, and like we talked about, and we've talked about the monosaccharides, the disaccharides, but you also have others, the polyols or alcohol sugar like sorbitol, mannitol, lactitol, xylitol, erythritol, isomalt, maltitol, and the oligosaccharides and the polysaccharides. So go through this and learn them. They may come in handy, and they will be important. So in next class, we shall be looking at different Nigerian dishes, and we shall be looking at their calorie content, basically how much calories are in these foods, and contributions from carbohydrate, lipids, and proteins. It is important to understand at this juncture that the three macromolecules, carbohydrate, lipids, and proteins, contribute to our caloric intake. And of course, what is calorie? I mean, to understand it clearly, calories are the things that make us fat. So carbohydrate, lipids, and proteins contribute to our caloric intake. So that is why in making a healthy decision on my food and the combination of foods that I should eat, depending on my genetics, depending on my physical activity, it is very important to know the total caloric content of that food. And as you know, what contributes to calories? Carbohydrate, lipids, and proteins. But of course, we'll look into that in the next class.